Hello, I'm Priyanshi Chaudhary, a volunteer at shiviforensics, and today I will be discussing different methods of DNA quantification. But first, we need to understand what is DNA quantification. It is the process of determining the concentration and purity of DNA in a given sample. For example, in forensic science laboratory, when we get a biological sample to develop DNA profile from it, the first and the foremost step is to extract DNA from that sample, and the second step is to calculate the amount and the purity of the DNA. This particular step is called DNA quantification. The need for DNA quantification is because for further processing, the STR analysis and PCR amplification is done, and in these techniques, the optimal amount of initial input DNA is required to get optimal results. And for STR typing, the optimal amount range between zero point 0.5 nanogram to 2.0 nanogram. For getting its optimal amount, the further standardization of the input DNA sample is also done by using different methods. Like in case the amount of DNA is too little, then we have to concentrate our sample, and this is done by using low temperate DNA approach. For example, in PCR amplification, when the concentration of the initial input DNA is very low, then more number of PCR cycles are added for the amplification of the DNA. If we have too much DNA, then the diluted samples are made so that it can come in the optimal range. The different methods used for DNA quantification are: first one is UV absorbance. In this method, the instrument called UV spectrophotometer is used to record the absorbance of the DNA sample at the wavelength of 260 nanometer. This absorbance is corresponding to a particular concentration of DNA and help us determine the amount of DNA in the unknown sample. The next thing this method is used for is to determine the purity of DNA, which is done by taking the ratio of the absorbance at wavelength 260 nanometer and 280 nanometer. If this ratio range between 1.8 to 2.0, then the DNA is considered pure. If the range is less than 1.8, then it is considered to be contaminated with proteins. If it range more than 2.0, then it is considered to be contaminated with phenol residues. The next method is slot plot method. In this method, a specific radioactive or chemiluminescent probe is used. This probe is of 40 base pair and it bounds to a specific region of chromosome 17, which is T17Z1. Now, this chromosome 17 is specific to humans, that this specific region is specific to humans and some of the primates like gorilla and chimpanzees. So, in this method, what happens is this probe binds with the DNA chromosome 17 and give different signal intensities. As you can see in diagram, there are signal intensities of unknown sample in between two lines, and outer two lines are signal intensities of the standard sample. So because of in standard sample we know that signal intensities belong to this particular amount of DNA. The comparison is done between both to determine the concentration of DNA in unknown sample. Now, this method also have some of the limitations, which are because this comparison is done manually, it become a very lengthy process. It takes several hours to perform it. Second is, it is also not specific to humans only because, as I said, that some of the signals are also for the primates DNA. The next method is picogreen method, which was developed by Forensic Science Service. The main component of this method is picogreen dye, which is fluorescent and intercalating dye. So this dye bonds only to double stranded DNA, and when it binds to it, it gives fluorescence, which is compared with the standard sample to determine the amount of DNA present in unknown sample. Now, the limitation of this method is also that it is not specific to human genome only because it gives result for the total amount of double stranded DNA present in the unknown sample. The next method is aliquant human DNA quantification method, which was given by Promega Corporation. Now, this is the method which is specific to human DNA only because it is based on allo repeats. Allo repeats are the repeats present in human genome in high abundance. So, what happens is in this method, a probe is used which hybridize and initiate a series of enzymatic reaction, and the end of that series of enzymatic reaction is the oxidation of luciferin, and this oxidation produces some light, and this light is captured by an instrument called luminometer. Now this luminometer, it captures light, so it gets some signals, and now these signals and this light intensity is compared with the standard that has been prepared in the lab, and this helps us determine the concentration of human DNA in the sample. So this is a very important procedure, and this was used in laboratories for a long time to determine the human DNA concentration. But with latest advancement, a new technology called real-time quantification, quantitative PCR, comes in play, and this is the technique used in the laboratories nowadays also. So this was developed by Hugachi and co-workers at Citus Corporation in early 1990s. So this method, the name real time comes because it helps us to monitor the PCR process as it is happening in the real time. Like the number of reagents that are being consumed, the number of PCR products that are being produced, we can watch it happening in the real time, and that's why the term real time quantitative PCR is given to it. Now, this is based on the approach of Tagman assay or the use of intercalating dyes such as cybergreen, which is very specific for double stranded DNA. So, in basic PCR amplification, what happens is there are three steps. First one is denaturation, in which both the strands got separated. Second is annealing, in which the forward and reverse primer got attached. And then third one is extension, in which DNA polymerase extend the both primers by adding the NTPs on it. Now these three basic steps are common in real-time PCR also. But there comes a different thing, a very specific, which is Tagman probe. As you can see in first diagram, there is Tagman probe. On its fifth hand, we have reporter dye. On its third hand, we have questionnaire dye. And this is at the time of annealing, that it is also attached to the end of the desired sequence that we has to amplify. So now extension will start, and Tagman DLA polymerase will start getting show extension, and it has a very unique characteristic that it chew off any of the sequence that come in its way. So when it will reach the end, it will chew off Tagman probe. And when it chews it off, the reagent reporter dye gets separated from quencher dye. Now, reporter eye is also unique because it has a characteristic of showing fluorescence. But in first step, you can see it is not showing any fluorescence because it is in proximity with quencher dye. That's why it is not showing any fluorescence. Now, in second step, you can see because the Tagman probe gets choose off, the reporter dye gets separated, it is showing fluorescence, and this fluorescence is recorded, and this is used to determine the concentration of DNA in the unknown sample. Now this whole process is being cleared. So how do we actually determine the concentration of DNA? So what we do, we see this graph that you are seeing on left hand side, and this helps us determine the concentration of DNA in the sample. And how it helps us, because this graph has three phases: exponential phase, linear phase, plateau phase. Now in exponential phase, what is happening, the new PCR products are being formed because this is the initial phase. So the reagents are being used, and almost all the reagents are producing new PCR products, so efficiency is almost 100%, and applicons are being doubled with each cycle. The number of cycles that are happening, the number of applicons are being doubled. So this is the exponential phase. This has highest efficiency. There is like the graph between the cycle number versus the log concentration is almost linear. It is very consistent in this phase. The next phase is linear phase. Now in this phase, what happened? The amplification is happening, but the PCR reagents are being depleting. Matlab, they are being used up, so they are depleting, and the amount of PCR product that are being produced is changing. It's not consistent in this step. So that's why the this step is not reliable enough for us to produce the results for DNA concentration. The next step, third step, is plateau phase. This is the final phase. In this phase, almost all the reagents are being used up in the that are being used to produce PCR products, and almost it is the end of the cycle. So this phase is also not reliable for us to perform and determine the concentration of DNA. So the phase which is used to determine the concentration of DNA is exponential phase, because it gives us the linear relationship between the cycle number and the DNA concentration. It shows the highest efficiency. That's why this phase is used. But only this phase is not used to determine the concentration. There is one more thing which is used, and that is CT value. Now, how do we determine CT value is there is a threshold line given by the software. Here you can see that threshold line. Now, just below that threshold line is also a line which is called baseline line. Now, this baseline, in the starting of the cycle, like in the starting of the PCR, some of the cycles are taking place and the PCR product are being generated, and as the products are being generated, fluorescence is being shown, but that fluorescence is not high enough to being recorded. So what we do, we basically cut, like we neutral that fluorescence and those PCR product and create a baseline. Afterwards, the number of cycles start taking place and number of fluorescences being shown which are significant enough, and then the graph starts to take a form. Now, the point at which the this line, this number of cycles cross this threshold of the fluorescence, that point, that number of cycle is called the CT value. Now, this CT value is very important because this is the point at which fluorescence exceed the set threshold, which was set by the software. Like the software set the threshold to be 0.2. So it is the point, that CT value is the point at which that uh, 0.2 threshold is being uh, exceed. So what happen is, see how CT value help us determine the DNA concentration. If we have lower CT value for any of the sample, then it means that the higher concentration of the sample which was extracted from biological sample was high, because it only take, if the threshold value, let's assume it's 15, and we have CT value which is lower for uh, B, and which is higher for, lower for A, which is high and higher for B. So for A, it could be like that number of cycles for it to take to exceed the threshold is approximately 2 or 3 maybe, or 5. But for B, it could be up to 10. So what it says that the initial amount of DNA would be higher. Otherwise, how can it exceed the threshold value within few number of cycles? So that initial input DNA would be higher. That's why it is crossing that threshold within few cycles, because the PCR projects that are being produced are higher than the other. So that's how it helps us in determining the concentration of the DNA. So both exponential phase and CT value are the most important component for the determination of the DNA concentration in the unknown sample. The most latestly used commercial kits for DNA to assay human DNA and Y chromosome DNA quantification are Quantifiller, Quantifiller 5, and Quantifiller Duo. And this is the method which is being used in laboratories nowadays. This is the most commonly used method because it helps to do DNA quantification and amplification at the same time. So it is also not time consuming. It is efficient to perform it at the same time. And if our quantification is not enough, then we can increase the number of cycle and we can adjust it. That's why this is the method which is very which is of high efficiency. Now, this is the end of the different methods that are used for the DNA quantification purpose, and thank you.